Thanks very much. Thanks for the invitation to come and speak with you guys today. Um, so as Anna mentioned, I know that the papers that I assigned some months ago were about SARS-CoV-2 and work that we've done on that in the lab. And we've certainly thought a lot in my lab about the processes by which new variants of SARS-CoV-2 arise and begin to circulate. But as the COVID-19 pandemic has begun to wane and other potential pandemics are on the horizon, we're beginning to think more about how to prepare for the next viral pandemic. And so one thing that's very much on our minds with regard to that is H5N1 avian influenza. So I will note that even though this is not necessarily as much in the news as it was, over the past summer when people were talking about the widening outbreak of H5N1 among dairy cattle, it's still very much a live story. In fact, so much so that between yesterday, when I thought I'd finalized these slides, and today, both stories that I put here have had new developments. So Mexico announced its first human case of H5N1 just a couple of days ago. And as of this morning, they announced that the child who was infected with that virus in Mexico, with no known exposure to dairy, by the way, we don't know how this child became infected. She has unfortunately died in the last 24 hours. And similarly, I was tabulating all the known dairy farms that have had outbreaks of H5N1 infection in the United States. And as of yesterday, the number was 998. And just today, we hear that there have been two new detections, one in California and one in Nevada, that brings the total number of infected dairy farms to around 1,000. So this is a story that continues to develop. That's 1,000 infected dairy farms. We don't know the actual number of infected dairy cattle. So just to try to tabulate some of these things from available public data from CDC and USDA. And by the way, one thing that's been made it difficult for us to track and control this dairy cattle outbreak is that there are multiple federal agencies who have some level of jurisdiction over this situation. CDC deals with human pathogens, USDA, APHIS deals with agricultural pathogens, and FDA deals with food animals. And so they all have some jurisdiction here, and it's been difficult for those agencies to decide who's going to do what. Even 1 year into this outbreak. So as of today, we know of 1,000 cases in dairy herds. This is in 17 different states. No infected dairy herds in the state of Wisconsin that we know about yet. And there's little data that we have on prevalence within herds, although we think that once the virus enters a dairy herd, the prevalence is probably pretty high. So that means a lot of animals on the farm probably get infected in short order. This is primarily an avian virus, so I should underscore that before March 2024, when we recognized that H5N1 influenza viruses were infecting cattle, it was thought that influenza A viruses didn't infect cows at all. So this was not on my pandemic bingo card. What we do know is that H5N1 viruses are highly pathogenic in poultry. And so there have been almost 1,700 recorded outbreaks of H5N1 infections in poultry farms in the United States in the past few years. Over 100 million birds have been affected. These have occurred in all 50 states plus Puerto Rico. And there's been extensive evidence of spillover of viruses infecting dairy cattle, then causing outbreaks on poultry farms. So the virus is being circulated between cows and poultry, and then infecting humans who are exposed either to cows or to poultry. So associated with this dairy cattle outbreak, we now know of 70 human infections in 13 different states. There's been one fatality in the United States and another in British Columbia. Interestingly, neither of those fatalities was associated with consumption of or exposure to dairy. So the virus that has been infecting dairy workers has caused like severe conjunctivitis, a viral pink eye, but we have no evidence that it's been transmitted from one person to another in any case. And there have been no fatalities or hospitalizations associated with infection with the dairy cattle virus. However, there have been these fatalities associated with infection with other H5N1 genotypes. We don't know the genotype of the virus that infected the child in Mexico, but I suspect it won't be the dairy cattle one either. There have been a number of infections of other mammals. So in the background of all of this is a global panzootic, that is a pandemic among animals, of a genotype of H5N1 viruses that seems to have a really unusually good ability to infect mammals. And so there have been 132 known infections of domestic mammals associated with this dairy cattle outbreak, 457 wild animals infected with viruses of similar genotypes, and that's over 39 states. Most notably, we've seen infections in cats that lead to severe neurological disease and death. Sometimes we assume from consuming infected milk on dairy farms. Okay, so wild birds continue to introduce new genetic lineages of H5N1 viruses into humans and other animals. And so today I wanted to look back at work that my lab has been doing over the past several years to try to understand the evolutionary processes by which influenza viruses jump species. So how do we get pandemic influenza viruses? So I'll review sort of the data that caused us to think about this the way we do, and I'll end with some brand new data. Okay, so a question on everyone's minds is, will H5N1 viruses cause a pandemic? And I think you could actually ask this question a slightly different way, which is, why haven't H5 influenza viruses caused a pandemic already? So if there are all of these viruses circulating in domestic mammal species with which humans have extensive and prolonged contact, and we know that infected dairy cattle shed infectious virus at extremely high titers in their milk. So like 1 billion infectious particles per milliliter of milk. That's a lot. Why has there not been more evolution towards a virus that can transmit easily among humans? Why hasn't there already been a pandemic? So that's the question I will try to address as we go along today. So a basic outline is as follows. We'll talk about RNA viruses and their ability to evolve rapidly. I know I talked about the paradox of scale, which is something that we think about a lot, but I'm going to focus a lot here on what happens within individual hosts and then when viruses are transmitted from one host to another. So in order to understand this, I'm going to have to give you some background about where pandemic influenza viruses come from, as far as we know, what forces shape viral evolution within and between hosts. So what happens in one infected individual, what happens when a virus goes from that individual to another. I will argue that transmission bottlenecks are an important factor that was previously underappreciated in affecting the trajectory of virus evolution. And then I will sort of pose a question at the end, like, what if avian influenza viruses are good enough at replicating in mammalian hosts? And what does that mean for the danger that they will cause a pandemic? I'm not trying to argue here, by the way, that I'm not previewing an argument that like we shouldn't worry about avian influenza viruses, nor am I saying that a pandemic is about to happen. I legitimately don't know. That is the nature of pandemics. Okay. So a big question that we have, and something that we tried to get at in the review paper that I asked the students to read, is how are within host viral evolutionary processes linked to global processes? And we've begun to think about that a lot in the fields of influenza virus and SARS-CoV-2 evolution in the last 5 years or so, because we've begun to realize that when you look at the global level, which is depicted by this cartoon here showing H3N2 seasonal influenza virus evolution over several years from the next strain group that's really great at visualizing these trends. When you look at the global level, you see that antigenic evolution of seasonal influenza viruses infecting humans is like almost deterministic. right? We have to update the flu vaccine every year or so because of the unremitting antigenic changes driven by natural selection, right? The selection of our antibodies on circulating viral strains. So that's almost a deterministic process at the level of the human population in the world. But the more we look at individual hosts, individual infected humans or animals or whatever, this is where the genetic variation that ultimately circulates worldwide arises. But we're finding that it's really hard to pinpoint the process by which a new variant might arise in one individual and then go global. How does it survive and get to the next host and the next one after that? So we want to integrate our ability to understand across the individual scale to the global scale. And hopefully through doing this, we can improve our ability to predict trends in global viral evolution, because that global sampling that we do is sort of inherently backward looking. Like, we can explain how viruses came to be the way that they are and why this clade or another might be being selected for at the human population level. But it would sure be great if we could sample a tractable number of individuals and then try to forecast what trends in viral evolution will come from that. Okay. So we know that RNA viruses have very high mutation rates. So this is a figure that is reproduced in textbooks and review articles and all sorts of things. And so this just shows genome size of various different types of organisms on the x-axis, and their inherent mutation rate, like the error rate, on the y-axis. And RNA viruses are shown in that sort of yellowy color there. And they have a very high mutation rate, as you see, and relatively small genomes. So we know that the RNA-dependent polymerases of RNA viruses almost all lack error proofreading capabilities. So they have a high error rate, so they generate mutations pretty quickly. And then RNA viruses have fast generation times, so maybe on the order of like 8 hours for influenza virus. They can replicate to very high titer in infected hosts. They can infect a very large number of hosts in a short span of time. And so you add that all up and you get high annual substitution rates. So that means as we sample viruses over time, we see relatively quick accumulation of consensus changing mutations in viral genomes. So that is just sort of basically true. And you have a lot of raw material for evolution. How does that relate to the emergence of pandemic influenza viruses? So in order to understand that, we have to look at another figure that I intentionally took from like a very old article that has been reproduced in many places too. And this just sort of shows, like, I don't know, the wheel of influenza with aquatic birds at the center. Right? So what we're showing here is many different species of animals that can be infected with influenza viruses, but all known influenza diversity, asterisk, influenza A virus diversity comes from aquatic birds. They are the reservoir of almost all known influenza virus genetic lineages. And you see that asterisk there. And I put it there because there are a couple of very weird influenza viruses that are found in bats and in no other creatures. But let's not let them ruin this basic idea. And so classically, we are told that domestic poultry and pigs are the most common and most important proximal sources of new influenza viruses infecting humans, right? And so we can see that here. Let's try this build one more time. Oh, geez. Well, anyway, you get the idea that viruses are coming from poultry into humans or from pigs into humans. Okay, so we can get into that if you have questions about it a little bit later, but that is the sort of classical understanding. Pigs are highly susceptible to influenza A viruses of many types, and humans are exposed to them as they are to domestic poultry. This is a cartoon of an influenza A virus particle. And what I want you to notice is a couple of things. So this is an enveloped virus, so that sort of purpley circle there represents the lipid membrane, the envelope of the virus. That is decorated with two main proteins. In purple, we see HA, hemagglutinin, which is the viral attachment protein responsible for attaching the virus to host cell receptors. So antibodies against that HA could prevent the virus from successfully attaching to a cell and getting inside it. There are 19 known different types, subtypes of HA defined by serology. NA, or neuraminidase, in yellow, is sort of a viral deattachment protein. It helps the virus free itself from infected cells when it's leaving the cell. There are 11 known serological subtypes of NA. And so I tell you that because H5N1 means that that is a virus that has hemagglutinin type 5 and neuraminidase type 1 in that virus. And theoretically, we could get influenza viruses that combine any of the 19 HAs with any of the 11 NAs, at least theoretically. What I also want you to see here is that there are 8 little squiggly lines which represent the RNA gene segments, all 8 of which must be packaged into a virus to make it infectious. So altogether, those 8 gene segments encode, depending on who you talk to, somewhere between 10 and 15 known proteins. So there's 10 principal proteins and some other accessory proteins. And that's what the virus needs to do to take over cells and make more influenza viruses, which are all influenza viruses are any good at. Okay. So the existence of these 8 different gene segments means that if you infect one cell with 2 genetically distinct viruses, you can, at least in theory, have 2 to the 8th, which is 256 different genetically distinct viruses come out. So you can have the 8 gene segments reassemble in any combination from this co-infected cell. And so this process, which we call reassortment, is a way for influenza viruses in nature to exchange genetic information and give rise to new combinations of viruses, some of which may be infectious to humans and not and evade our pre-existing immunity. And so we think this is what happens when pandemics occur. We get a reassortment event that introduces some genetically new gene segments into human infecting influenza viruses, and then we're all kind of susceptible to that. Okay. So in order for there to be a pandemic, there's a number of criteria that have to be met. So first, the viral polymerase complex that copies the viral RNA. That's made up of these 3 proteins that I list up here, PB2, PB1, and PA. That has to adapt to the human respiratory tract temperature, which is in the upper respiratory tract, we commonly say it's 33 degrees, and in the lower, it's 37. Whereas in the avian alimentary canal, where the virus typically replicates in aquatic birds, the temperature is a little higher, 42 degrees Celsius. And there are other host factors that the polymerase complex has to interact with. And so it has to gain the ability to do these things at human body temperature. And generally, that means losing the ability to do them efficiently at avian body temperature. Similarly, avian viruses need to acquire the ability to bind human-type receptors. So all influenza A viruses, again, with a little asterisk that we're not going to get into, for the purposes of this talk, all avian influenza viruses attach actually to sugar moieties, not to a specific protein, but to sugars, glycosylating proteins on the surface of an infected cell. So the HA of human viruses looks for sialic acid linked to galactose in what we call an alpha-2,6 configuration on the left in blue. Whereas in birds in the GI tract, they have galactose linked to sialic acid, but primarily in an alpha-2,3 configuration. So that's biochemically distinct, and influenza viruses can tell the difference between alpha-2,3 and alpha-2,6 linked sialic acids. So in order to transmit among humans, the virus needs to be able to bind those sialic acids that are present in the upper respiratory tract of humans, and they're in this alpha-2,6 configuration. There's other things that viruses have to do too to really efficiently transmit from humans, between humans, and really like become like a human virus, but we're not going to worry about those other things too much right now. So about 10 years ago, Yoshi Kawaoka's group here at UW-Madison and the group of Ron Fouchier in the Netherlands independently started doing experiments aimed at addressing the question, well, how many mutations do you need to convert an avian virus to one that would be transmissible in mammals. And to do this, they used a ferret model because ferrets are the sort of gold standard model for both transmission studies and pathogenicity studies of influenza virus. Their respiratory tracts are very similar to those of humans. And so for the purposes of studying flu transmission, they're a really good model. They're very susceptible to that transmission. Okay, so there were two papers that were in the news quite a bit several years ago. And they basically showed that a handful of mutations changing those two main functions that I talked about, the polymerase and HA, were enough to take an avian virus that is not normally transmissible between ferrets and make it transmit efficiently. In fact, in the case of Yoshi's group, they found that as few as 4 mutations conferred the ability to transmit between ferrets to an otherwise avian virus. So that's a very small number of mutations that you need, right? That's kind of worryingly small, I would say. So the next question is, well, what is the likelihood that human adapting variants could be generated and selected and transmitted in the course of a particular infection? So to address that question, we first asked, what is the genetic diversity in acutely infected hosts, right? So we want to understand how much, given that high mutation rate, how much variation can a virus generate over the course of one infection? So here's just an example of what a respiratory virus might look like in terms of genetic diversity in an acutely infected human. Here we're looking at a human-adapted virus, SARS-CoV-2, infecting humans. So this is from, I think I assigned this paper, from my PhD student, Kat Braun, and former student, Louise Moncla, led this study in PLOS Pathogens, where we deep sequenced SARS-CoV-2 viruses acutely infecting people. And we just asked, okay, how many different within host mutations, intra-host single nucleotide variants, or iSNVs, do we see in people infected with SARS-CoV-2? And the answer is there's a range, but generally speaking, not many. So the vast majority of infections had either no detectable mutations at all within that infected individual, or 1 or 2. That's very few. So that's not a lot of within host genetic diversity. And furthermore, on the right, we can see that when we just ask, okay, of the mutations that we observe, what proportion of them are at low frequency and like at various different frequencies, from 10% of the population of viruses that we sequence up to 50%, you see that the vast, vast majority of these mutations that are present within hosts are at very low frequencies. So our threshold frequency is 3%. So somewhere between there and 10%, vast majority of mutations are there. So what mutations occur are also at very low frequency. So this is consistent with the idea that most random mutations within this human-adapted virus are deleterious. They occur, but they're rapidly purged from the population by purifying selection. Okay, so now let's look at the same sort of metric, but let's consider an avian virus either infecting birds or infecting humans. So we were able to analyze high-quality deep sequencing data from H5N1 infected ducks and humans that were part of the same large outbreak in Cambodia. And this is work led by Louise Moncla when she was a student in my lab, and then continuing onwards after that. And so this is the same thing. We're showing iSNV frequency on the x-axis, and then just a slightly different y-axis, but you get the same idea. And so here, when we break out mutations by whether they change amino acids, non-synonymous, or whether they are synonymous, it doesn't matter actually. And we find that the iSNV frequency within infected humans and ducks also tends to be very low. So if there were diversifying selection, if there were selection for variants arising in those humans that helped adapt those viruses to humans, then we might expect to see more of a smile pattern here, where a handful of mutations at least go to high frequency because they help the virus replicate in humans, right? But we don't see that. And in fact, the mean frequency of within host mutations in humans and ducks is about the same. So we don't see evidence that selection is increasing the frequency of human adapting mutations when humans get infected with these avian viruses. Okay? So the same pattern applies here as we saw in a human-adapted respiratory virus in an acute infection. All right. So then we can ask, well, all of the mutations that we see, like, could we at least say whether some of those mutations occur in functionally relevant sites? So would they be predicted to change the phenotype of the virus at all? So when Louise mapped all of those mutations that we saw in humans and ducks in that Cambodian outbreak, she broke them into the various coding regions. So the 10 principal open reading frames of influenza A virus, they're shown here. You can see we detect mutations in all genes in the genome, in all gene segments, in humans and in ducks. But now let's leave highlighted only the sites that have some sort of known function according to the literature. And so now we've whittled it down to really just a handful of mutations that have previously identified functions, like functional phenotypic impact, that could potentially adapt these viruses to better replication in humans and other mammals. And one thing you might notice about that is that almost all of these mutations are at extremely low frequency, even though they have a putative beneficial effect in humans, right? So we'll zoom in just on PB2 and HA, because those are proteins that have well-characterized mutations that are really, really, really functionally important for adapting avian viruses to humans and other mammals. And so, for example, we see, let's see if I can do this here. Can you see my pointer? No. All right, I'm just going to walk over here. So this mutation that was originally identified actually by Yoshi's lab, called PB2E627K, is known to dramatically enhance the ability of the avian virus polymerase to work in human cells. And it's really important for both pathogenic potential and transmissibility of avian viruses in humans. So this is a mutation that was detected in a human, but at extremely low frequency. Okay, and we see some other potentially functionally important mutations in PB2 that would enhance its function in humans, but all at pretty low frequency. We can say a similar thing about HA. So there are mutations at a site, Q238, in two different individuals. One had an arginine and one had a leucine at this position. That is an amino acid that, that Yoshi's group and others have shown is important for changing the receptor binding specificity of HA. But again, we see it in a couple of individuals, does not become fixed, right? So altogether, this and other data show us that there's a handful of polymorphisms in these infected humans that are detected at functionally relevant sites, but their frequencies are low. We see very few. So out of 6 patients that we sequenced virus from, there were potentially functionally important mutations in 4 of those 6, but there was only 1 mutation that occurred in more than 1 person. So if you're a virus trying to adapt to humans, like, that's a pretty poor showing, I would say. Not to be judgmental. Okay, so do we have any other evidence that the mutations that we detect in these individuals could actually encode real phenotypic differences? Up to now, I've just been showing you, cDNA sequence, and we are predicting functions based on literature. What if we do actual assays? And so to address this question, we actually collaborated with Yoshi's group, and they had access to samples from a different cohort. So these were 7 patients infected with H5N1 viruses in northern Vietnam over a span of years. There's one from 2004 and others from a bit later. At least 2 clinical specimens were available from each of these people. And we sequenced directly from those specimens with no virus culture. So we should have a pretty good insight into what is the actual variability within those individuals. So in this experiment, we called within host variants that were present at least 1% of sequence reads. And then heroic efforts in Yoshi's lab were led by Hirotaka Imai to characterize any variant that was present at or above 5% frequency for its functional impact. So this talks about like multiple assays for HA binding and like, does it bind avian or human receptors? The stability of the HA molecule, which we know is important, polymerase activity assays, and all sorts of other stuff that Hiro led in Yoshi's lab based on our sequencing. So here's a slide from George showing you, George Dennis in my lab, who did all of the sequencing analysis, showing you that, again, we detect variation, particularly in infected humans, in every main influenza gene segment. There were sequences available from chickens as well that were collected contemporaneously, but not part of the same outbreak. So we see some mutations in those viruses as well. But when we take out those individual variants and, for example, characterize their sialic acid binding preference, here for reference, so this is a glycan array that presents these sialic acids linked to galactose in either alpha-2,3 bird or alpha-2,6 human configurations. But like they could occur on all different types of glycans in all different types of configurations. Some are long, some are short, some are branched, some are linear, right? And viruses like have preferences for different configurations like that. So if you spot an array of different glycans onto a plate, then you can flow HA molecules over them and ask which ones do they bind. And so here we're just going to pay attention to, like, if it's blue, it's bird, and if it's red, it's human. Okay, so here's a human virus, and you can see that its HA primarily binds to alpha-2,6 red sialic acids. But when we look at a bunch of different HAs isolated from humans with these different mutations, most of these had no known, no previously characterized functional impact. Just choosing 6 abundant, like relatively abundant, within host mutations in HA, we see almost no binding in the red column and a lot of binding in the blue column, right? So that tells us that these mutations that are arising in HA in humans infected with H5N1 viruses, like, yeah, they're mutations, yeah, we can detect them, we can make those HAs, but they do not confer the ability to bind human receptors. These HAs stay avian, okay? So Hiro also wanted to do a different way of asking this question. And so he used human tracheal explants and then took virus, infected those tracheal explants, and then asked, can we stain for virus infecting these cells on the tracheal explants? And so the tracheal lumen is at the top of these images, and then the reddish brown is going to be a viral antigen that we detect with immunohistochemistry here. And so what this is showing you is that in this panel N on the left, you've got a human pandemic virus, and that lights up this trachea, right? So there's all kinds of virus infecting this trachea. And then there's one mutation, this one in the middle, that gives you a number of brown cells in this human trachea. So like maybe that enhances the ability of this avian HA to infect human tracheal explants. But most of the mutations that we looked at, so when we make an H5 HA that has this 203L mutation on the far right, you see almost no brown staining at all. So this virus does not do a very good job at infecting human trachea, human upper respiratory tract tissue. Okay. So what I've shown you through genetic and phenotypic characterization is that sometimes mutations that are theoretically beneficial to the virus in terms of enabling it to replicate better in human cells, sometimes we detect such mutations in H5N1 viruses infecting mammals, infecting humans. But they remain at low frequency. And the phenotypes of these viruses remain remarkably avian. So avian viruses infecting humans, even when they cause severe disease, even when these people have passed away, the viruses stay avian in genotype and phenotype. So then we'd like to know, well, if these viruses were able to get out of that host and into another one, what would happen? What happens to viral diversity? So if we go back to SARS-CoV-2 and just characterize a human respiratory virus infecting humans, and I should say, like, the data that we see for SARS-CoV-2, we have similar data for influenza, but it's not as rigorous. So I'm going to show SARS-CoV-2 data. So here we're just asking, when we see within host mutations in SARS-CoV-2 infected people, so that's the frequency of ISNVs in the index on the x-axis. So what is the frequency of that same mutation when we have a paired contact? So we know that the index patient transmitted the virus to a household contact. So what is the frequency of the mutation that we see in the index when it gets into the contact? And what this shows you is in the upper right up here, that indicates that a lot of mutations that were at high frequency in the index patients, they are also at high frequency in the contact. Okay, that makes sense, right? And then mutations that were present at low frequency in the lower left here in the index, they are maybe detectable in the contact, but often not detectable at all. So they're lost when we transmit to the contact, right? So that all kind of makes sense. We think that it takes maybe 15 to 50 virus particles to found a new infection. That's not a lot of genetic diversity. That's a transmission bottleneck. And indeed, when Louise modeled the transmission of variants as a Poisson process, knowing the substitution rate and serial interval of SARS-CoV-2 infection, she estimated that the majority of transmissions would result in no change to the consensus sequence at all. So maybe every once in a while you get one consensus level mutation in SARS-CoV-2 transmission. So in typical acute infections, viral diversity is low and mutations are rarely transmitted onward. So now let's look at transmission of H5N1 in mammals. So to do this, we went back to the ferret study that I described earlier that Yoshi's group did and sequenced every virus we could from every animal in that study. And so to understand just how that study worked, you have an index ferret that's placed in one cage and directly inoculated with a very large influenza virus. And then one day later, you place an uninfected contact ferret in an adjacent cage that's about 3 feet away. So respiratory droplets can diffuse from the index to the contact, but they can't touch each other. And so then you ask if, when you have many pairs like this, what is the frequency with which we observe transmission from the index to the contact? Okay. And so what we wanted to know is even if the virus diversifies in the index animal, are we going to see a large amount of diversity transmitted to the contact, or are we going to see a decrease in diversity, maybe through selection or maybe just because of randomness, because of founder effects? So we quantified viral diversity using deep sequencing. So we use summary statistics to do this. One of the main ones we used is a statistic called pi. Anybody familiar with that? You people? Okay. So this is the number of pairwise nucleotide differences per site, and we actually can calculate pi s and pi n. That is like the number of non-synonymous pairwise differences and synonymous pairwise differences. And this is corrected for, so this corrects for the fact that there are more non-synonymous sites than synonymous ones. So basically, if the value of pi n is greater than the value of pi s, that could suggest diversifying selection in this population that you're looking at, whereas pi n less than pi s could suggest purifying selection. Does that make sense? Okay. So then what we did is we compared the values of pi n and pi s in index animals, so in all the index and all the contact animals by gene segment. And so when we look in HA, the attachment protein is encoded by HA, we find a statistically significant reduction in both non-synonymous and synonymous nucleotide diversity across the transmission bottleneck, right, for HA. Now, when we look at the other gene segments, interestingly, so this is NA and M1, the M1 gene, we find no such reduction. So there is a specific reduction in genetic diversity in HA associated with transmission of these avian viruses in ferrets. So to me, that suggests that transmission of H5N1 viruses in mammals is associated with selection on HA. So we are selecting for particular HA alleles that are good at transmission, and we don't care so much about which M or NA alleles they are associated with. So there is some level of selection that seems to reduce genetic diversity from the index to the contact. And those bottlenecks appeared to be selective in this experiment anyway. And so other things that we noticed that were interesting is that we did see in some animals the increase of particular mutations frequency over time. But when we look at the contact animal, we didn't see any kind of intermediate frequencies of these mutations. They were either fixed or lost after transmission, suggesting a relatively narrow transmission bottleneck. And particularly remarkable was this guy right here. So this mutation was present at around 6% of viruses in the index animal. And 2 days later, when the contact became infected, that mutation was fixed in the contact. So even very low frequency variants can transmit in this scenario. All right, so altogether, that says to me that the transmission bottleneck can dramatically reshuffle allele frequencies from the donor to the recipient. And if natural selection can occur during this process, well, then this is a way to kind of filter out variants that might be better at mammalian transmission. So that evolution may not be occurring over time in one infected individual. You know, a mutation arises and goes to high frequency. But if that virus gets out of that first individual into another, then you have the ability to select for mutations that confer better transmissibility. Okay, so to wrap up, I just want to bring this into the present. So this is work that we've done over the past several years with a number of collaborators. And now we're interested in what does it look like? What are these avian viruses infecting cows look like? What is their evolutionary potential? So Yoshi's group has employed their ferret model with these bovine-derived avian H5N1 viruses. And so one of the first viruses that they looked at was a particular isolate from a dairy cow in New Mexico. And so it belonged to the hemagglutinin clade called A2.3.4.4b. That's the one that's circulating worldwide. This genotype that we call B313, that is the total constellation of all 8 gene segments. That has a name, that's what it is. So this is like a sort of standard cow-derived H5N1 virus, now that there is such a thing. And so they asked, how well does this virus infect donor animals, index animals? And in 4 ferret pairs, how often do we see transmission? So in orange on the bottom, you see that a control H1N1 virus, that's a human virus that we expect to transmit in ferrets, that transmits 4 out of 4 times. Although this cow virus replicates to even higher titers in purple above in the ferrets that are directly inoculated, it does not transmit at all. Pretty interesting. Okay, so let's zoom in on those directly inoculated donors and ask, how diverse are the viruses infecting these animals? Do we see any evidence for diversification of viruses or the accumulation of mutations that we might predict to have phenotypic impacts? And this is work done by Heather Moscovich, who's a postdoc in my lab. So she visualized the data a little bit differently, but the same idea here. You have a heat map showing particular substitutions in the 8 gene segments. And so we do see accumulation of mutations in at least one animal in every gene segment. The inoculum virus was remarkably free of mutations, only one detectable mutation here. But again, these mutations tend to remain at very low frequency, and we do not see much evidence for emergence of mutations that would be predicted to have a big phenotypic impact. When we quantify the number of mutations that occur over time in these directly inoculated ferrets, That remains low, although by day 7, 2 of the 4 animals are accumulating more mutations. And then we've tried to visualize this a little bit differently, but just sort of looking, instead of the frequency bin that I showed you at the beginning, this is the same idea here on the right. We're just saying what is the cumulative proportion of iSNVs, of mutations that are either synonymous or non-synonymous. And so if there were positive selection, if there were diversifying selection, we might see non-synonymous mutations accumulating faster, and synonymous ones accumulating more slowly, and vice versa if we had purifying selection. But here you see they're accumulating at very similar rates. So to me, this means that there's neither strong purifying selection nor strong diversifying selection on this particular cow-derived avian virus in the ferrets. So we see little evidence for additional mammalian adaptation of this bovine-derived virus in ferrets, even though it doesn't transmit efficiently. So altogether, this says to me that once avian viruses find their way to suitable cells in mammals, they may be actually quite content to replicate, even to very high titer. And it may be very difficult for mutations to arise that adapt them to mammalian transmission and then have those mutations get out of those individuals. So some conclusions and interpretations of all this work. During typical acute infections, and this is a phrase I say a lot now because I think there's an important role of unusually prolonged infections. So during typical acute infections, within host respiratory virus evolution is constrained by randomness and purifying selection. Transmission bottlenecks, we've shown, can rapidly change allele frequencies. They could be random. Selection may occur during those transmission bottlenecks. This could be a mechanism that links evolutionary processes across scales by reshuffling allele frequencies. So I think it's important to understand how these bottlenecks work. Within host diversity of avian influenza viruses is low, even in humans that have been sampled many days or dozens of viral generations after they were infected. So these sort of stochastic random processes may substantially constrain the ability of avian viruses to effectively adapt to humans over the course of a single infection. But transmission might be the key. So transmission onward from that first person might be really important. Okay. So finally, some unresolved questions. What mechanisms govern these transmission bottlenecks? When are they selective? When are they random? What is the relative contribution of these processes? I think that has a lot of implications for like the size of the height of the species barrier. What situations promote the ability of new variants arising in one host to be able to transmit widely, ultimately globally? So is it the case that there's like evolutionary jackpots, like 1 in a million acutely infected hosts, like the virus happens to hit upon a particular mutation and that variant happens to be transmitted onward and that sparks a larger outbreak? Or what about the role of unusually prolonged infections in promoting the evolution of adaptive variants? So maybe it's not like one unusual jackpot in acute infection, but maybe there's a class of hosts who, in whom evolution can proceed a little bit better. So that's sort of what our review article was getting at. And then what is the risk that H5N1 viruses circulating in cattle today will cause a human pandemic? I wish I could answer that question, but I don't know. And finally, I think a super important question that I've not really talked about in this talk, because it's not really about science, but rather about society, is how prepared are we for a new pandemic? I would argue that the COVID-19 pandemic has actually left us less prepared to deal with a novel influenza pandemic than we would have been 5 years ago. And that's a problem. So I'll leave you with that thought. And I would just like to thank a large number of people in my lab, current and former, and in collaborators here at UW and beyond. So all of these folks have contributed in some way to what you saw today. And of course, all of our funders. Thanks so much for your attention. Yep. Yep. Yeah, so that's a good question and something that I decided not to go into in this talk. But so the question is, um, if there's such a thing as a human receptor and a bird receptor, how the heck do these bird viruses not evolve the ability to attach to the human receptor and yet infect humans? And so the answer is that we do have bird type receptors, humans do, as do pigs and other mammals. But in humans, they tend to be these bird-like receptors, alpha 2 3-linked sialic acids. They're in the lower respiratory tract. And so we think that that is why we observe high titer virus replication in the lower respiratory tract in people who are infected with H5N1. That could be a mechanism for the severe disease that's been observed previously in H5N1 infections, because lower respiratory tract infection is itself associated with severe disease. So maybe it's also why we don't see as many infections as you might expect with H5N1, because maybe you have to be exposed to a lot of virus and like breathe it way down into your lungs to get infected. This has been sort of the received wisdom. In the cattle outbreak, most of those people who've been infected have had conjunctivitis. And some have respiratory symptoms, some don't. So it turns out that your conjunctiva also have bird-like receptors. And so maybe that confines replication of those viruses to the spot where anatomically those receptors exist. And because it's not in the upper respiratory tract, that helps us from transmitting onward. Good question. So that's a good question. Basically, do age and sex matter as biological variables? Not sure we're allowed to talk about that anymore. But I would say that's not really been examined deeply in these studies previously. There have been interesting potential associations between like biological sex and severe disease for SARS-CoV-2. So men are at higher risk for severe disease, but we still don't really know why. Um, in in avian influenza infection, I mean, this is getting further away from your question, but like there are sort of age cohort effects in terms of susceptibility to different avian influenza viruses. And there's some really cool work from Jamie Lloyd-Smith at UCLA that suggests that like your birth year influences which viruses you are most susceptible to. And that's probably because you immunologically imprint on the first influenza virus you're infected with. And like that gives you a starting point of sort of immunological cross-reactivity with other viruses. And the further you get away from that, the harder it will be for you to have some cross-reactive immunity to these like weird avian viruses. So we can go into that if you care, but that's far from your question. Yes. Yeah, absolutely. So there are very, very highly conserved nucleotides at each end of each gene segment that are important for packaging. And so like there's like a whole field of study of like how does the virus know that it has 8 and only 8, right? And like there are combinations of gene segments that seem to like to package together, right? And so that's about as much as I know about that particular thing. But like these are the packaging signals that are used to ensure that you get 8 gene segments. Now, like it is not a perfect system, and so with like electron microscopy, you can observe viruses with like 7 gene segments and 9 gene segments. So it does occur, but you need 8 to be infectious. Yeah, that's a really good question. So basically, like, will different viruses have different transmission bottlenecks, like bigger, smaller, right? Um, I think that's possible. I suspect that it has a lot to do with the mechanism of transmission. And so I think about that in two ways. One, there's a little evidence from our lab and from Ben Tenuver's lab that that contact transmission is associated with a bigger bottleneck, like more different variants being transmitted than respiratory droplet transmission. And so you can imagine, like, if you are directly slobbering on somebody, like the infectious dose that you are giving them is much bigger than if they breathe in one tiny respiratory droplet that came out of your larynx, right? And so maybe then you preserve more diversity from the donor to the recipient. So that, like, transmission mode is probably important. In HIV, we think that, and like people have studied this in HIV for a very long time, so they're very interested in what they call transmitted founder viruses. And so through sexual transmission, we think that really one genetically distinct virus founds transmission almost all the time. And there's even a selection for viruses that can infect cells of a certain type. So there's a co-receptor that viruses need to use to get into infected cells. And over time in an infected individual, HIV can run out of cells expressing that co-receptor. And even so, when every time the virus is transmitted, even if the donor person has mostly viruses that bind what we call CXCR4 as the co-receptor, like always transmission involves viruses that use CCR5, a different co-receptor. And that probably has to do with the population of cells that is available to infect in, like, the reproductive tract mucosa. So there's, like, you have to know the system, basically. But yeah, I bet there's differences. Thanks, everybody.